Welcome to the Terminology Leading Practices 2023. My name is Jim Case, and I'm the Chief Terminologist at SNOMED International, and I will be moderating today's sessions. All questions will be answered at the conclusion of the presentation. Online attendees, please use the Q&A button to type your questions to the presenter, and on-site attendees, please use the microphone provided in the center of the room. I'm now pleased to introduce Sarah Warren from SNOMED International, who will be presenting Genus and Genius, a microbiology group. Sarah, please go ahead.
Thank you very much. So hello, everyone. I'm Sarah Warren, and I'm a terminologist at SNOMED International. My presentation is about the Microbiology Working Group. So the group was formed in October 2021, and I was asked to support the group and have been involved in their work ever since. So this presentation will cover the development and output of the group. It will cover the scope of the work, the type of requests that have come through, and letting you know the progress of the group along their work plan so far. So as with every presentation in Expo, I'll speak for about 15-20 minutes, and there'll be time for questions at the end. So in August 2021, members began to tell us that there was large amounts of microorganism content in extensions. This is high priority content as it's used in lab reporting, but it's also internationally relevant content. There's a high workload associated with the microorganism content in terms of maintenance and promotion. Organisms are constantly changing names, being reclassified. So there's quite a lot of time input to keep those up to date. And with the promotion of content, we're talking about hundreds of concepts, so it would take quite a long time to promote this to international. This links in with the service level agreement limits, which means that no more than 150 concepts of the same type can be promoted or requested per 6 months. So it actually takes quite a long time for all of these concepts to make it into the international edition. The problem with if extensions keep this content is if SNOMED International release duplicates, it then leads to inactivation in extensions. So there was a general call for kind of more collaborative working, for the sharing of content, and even questions around whether some of these bacterial updates or organism updates could be automated in some way. So the Microbiology Working Group was proposed. Information about this was circulated on the member forum confluence pages, and Ian Green took the idea to the senior management team who agreed for this task and finish group to be created. In October 2021, meetings began and kindly John Snyder stepped up to be chair of the group. And most importantly, it was agreed that the work from this group would not be subject to the normal service level agreement limits. So this was really going to focus on bringing in the backlog of organism content into the international edition. So this is the scope of the group, and it was to identify microorganism content in extensions that was eligible for promotion to the international edition, and to define a process for that to be promoted going forward. And just to note that organisms below subspecies level were out of scope for this work. So there's many countries involved at some point along the way. So the US, Canada, Estonia, France, and the Netherlands, Germany, Norway, Denmark, Hong Kong, and Austria were all involved in the online meetings. The countries on the top row were countries that went on to promote or request content through this group. There was also many teams involved at SNOMED International, so the content and mapping team, the implementation support team, customer relations leads, and external authors. So in October 2021, SNOMED International hired an external author and subject matter expert, Jeff Wilkie, to work on this project. Initial meetings began, and they were primarily focused on coming up with the scope of the group, the work processes, the work plan. But they also acted as a forum for extensions to bring questions about microorganism content to SNOMED International, and also for SNOMED International to give updates on where the work was at. At this point, the supporting references that would be accepted by SNOMED International were agreed, and John Snyder was able to develop an automated script to compare LPSN against SNOMED CT. So LPSN is our preferred resource for bacteria naming. And the group decided they wanted to incorporate this into their work plan. So I'll just talk a little bit more about the comparison script done by John. So this compared SNOMED CTs, this was the June 2023 release, against a downloaded flat file of LPSNs. This was done in July 2023. There was two outcomes to this script. So the first outcome was it found a concept in SNOMED CT where the FSN was listed as a synonym as per LPSN. And in those cases, it would recommend that the descriptions of those concepts were renamed. The second change it identified was it found two concepts in SNOMED CT. One concept would have the correct name as per LPSN, and the second concept would have an FSN as a synonym as per LPSN. And in these cases, it would recommend that the synonym concept's inactivated and the descriptions move to the current concept or the correct concept. There were a few limitations with the LPSN flat file. It wasn't the easiest to work with and didn't fully reflect the kind of format of the LPSN website. It didn't give any information on genus level. It only gave information of genus level and below, and it gave no information on the type of synonyms or whether those synonyms were validly published. The overall outcome of this work was very useful, though, and those changes were uploaded to SNOMED International via the content request service. So going back to extensions that were identifying content, microorganism content that they wanted to promote to the international edition, they looked at 5 areas, and the 3 areas they found content was bacteria, eukarya, and virus. So this was the general work plan. So they would promote the bacteria and virus content. That was chosen because bacteria had the largest numbers, and it's also got the best evidence base. So we're using LPSN as our reference for this. There would be a cutoff date. This would be the point after which changes would go back to the normal CRS request process. This was a task and finish group, an organism constantly changing names. There did need to be a date after which it would revert back to normal processes. They would then run the LPSN SNOMED CT comparison script and work on that. And after that, they would promote eukarya content. They would then review any further work areas before the group was discontinued. So how did this look in extensions? So they identified and extracted content, and this was done either with an ECL or an SQL transitive closure script. It was important that this was published content within the extension, but also validly published within the supporting references. And for each concept that they were pulling out to promote, they also pulled out the effective time for that concept. Once the extensions had done that, they merged all the content in the spreadsheet and duplicates were identified. If there were duplicates, it was the concept with the earliest effective time that would be the concept that would be promoted. And this meant that no NRC had an advantage over the other. The only exception to that would be if SNOMED International released the same concept in the time of this work, then the SNOMED International concept would be given priority. At this point, the references were checked to make sure that there were ones that were accepted by SNOMED International, and the concept was checked that it had the correct name or the current name as per the reference. If it didn't, this was updated in the CRS request and the concept was changed on promotion. After this point, the spreadsheets were separated back out to the extensions, and the individual extensions uploaded these to the content request service. And this meant that if there was any clarification questions, it would go back to the owning organization. For managed service customers with low numbers of content to promote, this was done through the functionality of the tooling. So the work at SNOMED International, so we received all the changes via the CRS requests, as this is our method of workload tracking. And we also received spreadsheets for the large numbers of requests. For managed service customers with the high numbers of content to promote, this was done by our tech team that moved that into our authoring platform for us to save time. The content was then authored and reviewed by Jeff Wilkie and myself before any normal clarification process through the CRS tickets. And of course, this is primitive content with no modeling. After the content it was released, all the CRS requests were then closed. So for primary resources, our full list of accepted references is available in the editorial guide. But for the 3 areas we were working on with this content, so for virus, we were using ICTV, bacteria content, LPSN, and for fungi content, we were using Microbank and Index Fungorum. So Microbank and Index Fungorum don't always agree, and that's usually if one of them has references that are out of date. And in those cases, we would look at those references, or look at recent journal articles to see what the current name of that organism is. Our ideal resources have clear editorial principles and are available to all authors. So although printed manuals are very good, they're not freely available for us to use. So where are we at? 100% of the bacteria and virus content has been promoted. So that was 735 changes. And this was done between November 2022 and July 2023. John then ran the LPSN SNOMED CT comparison. And we're about 15% of the way through that work. We've also just identified the eukarya content to be promoted, and there's 331 changes there. When John ran the LPSN SNOMED CT comparison, there was 964 changes. So this was slightly more than we were expecting to come from this work. And when we started on this, quite a few of the changes required more research. There was often the addition of more supertypes, and there's many classification effects on kind of disorders and procedures, which then needed investigation. So the group decided they wanted to pause the bacteria content update, go back and promote the eukarya content before going back and continuing with the bacteria content update afterwards. And this fit with the scope of the group, which is really to bring in the backlog of content from extensions. So just looking at the type of request that came through. So one of the biggest worries was there'd be quite a bit of duplication between extensions, but there was only about 1%, so it wasn't too bad. The change type of request, so most of it was the promotion of content, which is what we're expecting. Some were description changes. So this happened if an extension went to promote a concept, but found it existed in the international edition, maybe under a synonym. In those cases, they would request for the international concept to be updated rather than promote their own. There was 22 new concepts requested, and this came from BioMérieux in France, and these were submitted through the US NRC. This is the type of organism requested. So these are in number order, and the top three are species, resistant type qualifiers, and genus concepts. And this was the outcome of the LPSN SNOMED CT comparison script. So this was finding two changes. Most of the changes were just for the renaming of organisms in the international edition. And there was 287 identification of two concepts where one would need to be inactivated. And simple's highlighted there because, as I said before, when we started working on these, they actually took a little longer than we were expecting. So for the eukarya content that we've just identified in extensions to bring in, again, in number order, the top two are species and genus concepts. There was a similar amount of duplication in this area of content with about 1.5% duplication between extensions. So there was many authoring discussions along the way, and it's planned for these to become editorial guideline changes to make kind of the addition and maintenance of organism content easier in the future. So I'll just talk through these. So the top one relates to bacteria content, so specifically for LPSN as our reference. So we had been adding homotypic synonyms to content, but early on we decided that we would be adding heterotypic synonyms to concepts. There was 5 X-like organisms where X is kind of a genus or a species name. These are very difficult to add. It's often difficult to find consistent references that define the meaning and the use of these type of organisms. Hyphens aren't used consistently, which makes it very difficult to work out the correct place for these to be, and also difficult to pull out where the research for these kind of organisms. So we had to agree the format for these before they were added. Organism groups and organism complex, often these are interchangeably used, often in the same journal article. So we often didn't know which one to go with, but there didn't seem a particular reason why it was either a group or a complex for those. For orphan species, sometimes a genus would no longer exist, and in those, but not all of the children had been reclassified. And in those cases, we would have to reassign parents to those children as supertypes in the hierarchy. So the assignment of non-linear parents, often this would mean there'd be multiple parents to a concept. These are all stated relationships in this hierarchy, so there needed to be careful review to make sure none of those were missed. And organisms with qualifiers exist outside of established naming mechanisms. Again, when they're not named consistently in research, it just makes it more and more difficult to find consistent references for these. Microbank taxon synonyms. Microbank makes no judgment on the validity of these taxon synonyms, so we don't routinely add them. But if they are requested, we then require further journal articles that link that synonym with the correct name of the concept. With the LPSN SNOMED CT comparison script, with some of the changes, we were finding that the reclassifications didn't reflect the wishes of the clinical community. And in some cases, those documented pushback. So we held back on some of the changes in that area. So next step, so we're just in the process of promoting the eukarya content, so 330-ish concepts to promote there. Then we'll go back and complete the bacteria content update. After that, the group's going to review any further work areas that they wanted to do before the group is discontinued, and all requests would go back to the normal CRS request process. There's no official end date to the group as yet, but it's likely that this work will continue through 2024. So if you're interested in participating in the group, we're always welcome for new members. They have a microbiology project group on the Confluence pages on the member forum site. And if you're interested in microorganism authoring, further information is in our editorial guide. That's my email address there if you've got any questions on the presentation, or you feel free to send a query in via Freshdesk. So as in conclusion, the microbiology working group was brought together to address the backlog of organism content in extensions. It started in October 2021 and primarily focused on bacteria, virus, and eukarya content, but they also ran an LPSN SNOMED CT comparison script. After completion of the work, the group will discontinue and all requests will then go back to the normal CRS request process. So thank you very much, and I hope you enjoyed the presentation.
Thank you. Sarah will now take questions. As a reminder, if you're in the room, please use the microphone to ask your questions.
Hi, thank you for your presentation. It was really interesting. I worked with bioMérieux some years ago, and microbiology is one of the use cases in France, so the French industry will be happy to contribute to the group. Just a quick question. Are you planning to applying the same process that you have done with the LPSN, but with the ICTV concerning the various signings?
Is it in terms of the kind of comparison script?
Yeah.
No plans to at the moment. I think the difficulty is having the downloadable flat file to use in comparison. So I think that's the major limitation there. And as mentioned, we were finding that some of the changes had the kind of clinical pushback. So we really need to wait for kind of acceptance of those changes. And often the best way to wait for the acceptance is to wait for them to come through as requests to us from users. So to answer your question, no plans as yet.
Okay, thank you.
Yeah, thanks. That was really good. I think I had a lot of questions throughout it, but each of your slides addressed them all.
Okay.
But the only one I think that wasn't, and maybe I missed it, was for those things where you've got the microbial resistance sensitivities. Was that modeled as an attribute, or we just got simple is a
relationships? Yeah.
That's cool.
Last chance for any other questions. If not, we will conclude this session. Thank you, Sarah.